Good afternoon, everyone. My name is Elijah Baiceros, and thank you all for coming out to my presentation. Today, we will be discussing the effects of oxidative damage on the stability and processing of hdNA. First, let's review what traditional DNA looks like. This is the double-stranded helix that we typically learn about in high school and what we see in movies when they want to portray genetic material. But DNA can adopt alternative conformations such as hdNA, which is triple-stranded DNA. As you can see, there are two strands that are interwoven with a third strand that has decided to fold back onto the main duplex. So how exactly does this form? Let's walk through an example. So here we have a traditional double-stranded duplex of DNA. One side is blue and one side is orange. As you can see in this image, the two original strands are present, but the blue strand has decided to fold back onto the duplex. So how is this possible? Well, traditional bDNA only forms Watson-Crick base pairing, but hdNA utilizes both Watson-Crick and Hoogsteen base pairing. The difference is the position where the bases are hydrogen bonding. Again, as my colleagues have pointed out, the single-stranded DNA is also highly vulnerable. So why is this important? Well, previous literature has found that hdNA is inherently mutagenic in mammalian cells and that hdNA structures induce mutagenesis. This results in fragile sites or mutation hotspots that lead to lesions and eventually double-strand breaks. This is caused by the interaction of hdNA with polymerase enzymes during DNA replication. These double-strand breaks result in single-stranded DNA, which are more susceptible to damage and can lead to large deletions, which lead to overall genetic instability because the newly replicated DNA sequence is not what it was originally. And this can lead to disease. So this study aimed to answer the following question: are hdNA sequences hotspots for DNA damage? To do this, we focus on hdNA's interaction with induced oxygen species. So what is oxidative damage? Oxidative stress on DNA can come from endogenous reactive oxygen species that are products of daily cellular metabolic pathways. ROS are in the form of free radicals. In this example that I have listed, hydroxyl radical is there. ROS then leads to DNA damage. And it is important to note that oxidative damage via ROS is not random, and previous literature has found that ROS prefers sequences that are hotspots for hdNA forming sequences. If DNA damage from ROS is not repaired, it can lead to point mutations via transversions, which cause overall genetic instability and disease. ROS damage mainly results in point mutations, which will be the focus of this. So among the four DNA bases, guanine has the lowest redox potential and is the most easily oxidized. This means that in the presence of endogenous ROS, it is likely that the guanine will gain an oxygen at the 8th carbon to become 8-oxoguanine. 8-oxoguanine is often used as a biomarker for oxidative damage in the cell for this reason. If not repaired, the NER are then able to come in and remove damaged bases. However, these pathways lead to abasic sites. On the right is an example of a nucleotide that is missing its base. So based on previous literature, we know that hdNA leads to large deletions and that ROS damage on bDNA mainly leads to point mutations. So we believe that hdNA with oxidative damage is repaired similarly to hdNA without oxidative damage. hdNA structure will be what the repair pathways mainly identify, possibly causing more large deletions. So this study analyzed data in two different ways. First was in DNA sequencing to generate mutation spectra that were used to determine how the DNA was being repaired, the HDNA and the bDNA. And second was collecting thermal melting studies, melting points to determine if the triplex even formed and the stability of the triplex when compared to control groups. Both studies examined the effects of ROS on the HDNA. Data collection was performed by a previous capstone group and Dr. Sewell-Foote. In short, HDNA plasmids with and without oxidative damage were transfected, then transferred, and finally isolated to be sent off for sequencing. Once the sequencing happened and this data was collected, the sequence was provided and the chromatograms were also provided. What Curran and I, my lab partner, over the summer had to do is go in and manually insert which unknown bases that the machine could not read at some points. And this enabled us to compare the original sequence to the experimental sequence. APE and BLAST were used to perform these methods. So the purpose of that procedure was to determine the types of mutations in the sequence to speculate which repair pathways were involved. Here we see an example of a perfect alignment, which typically doesn't happen, but all the bases in the original sequence are aligned with the experimental sequence that was replicated and repaired. Here we see an example of a large deletion. As you can see, the bases in the experimental sequence are not even present, indicating large deletions. These were defined as, large deletions were defined as 20 to 300 base pairs missing. And last is an example of a point mutation where the C has now, my mouse is not clicking, is either a T or an A. This analysis was then overall quantified to produce this data. So some of the results that we saw from this were that one, HDNA has more large deletions, as you can see in this yellow-orange bar, and that damaged HDNA has even more large deletions in the presence of oxidative damage from ROS when compared to the HDNA that did not have ROS applied to it. So what does this mean? Well, it might indicate that different repair pathways were involved. And specifically, HDNA with ROS might still get NER repairing, which is leading to the great increase in large deletions. So in part 2, we wanted to evaluate how chemically modified bases, adeoxyguanine and abasic sites affected triple-stranded DNA. And so here is an example. Here in the first example, we see a triple-stranded foldback in which adeoxyguanine is in the middle strand. We obtained data for the adeoxyguanine mutation at 4 different locations in the triplex sequence, and for 3 out of the 4 locations for the abasic site. For the thermal studies, raw data was collected by Drs. Sewell-Foote, Sarah Coe, Livy Drummond, and Rachel Frey using the Carey 100 UV-Vis with a temperature controller. This raw data was then used to generate a temperature versus absorbance curve to identify the melting point. In this example, OG1 and AB1 are compared against the control, and we can see a shift to the left, which indicates that they are less stable. This graph could also be generated for all 7 sequences if we wanted to see that. The point of inflection, in this case, is the melting point, and to quantitatively calculate this, the first derivative graph was generated, and the highest point from this graph was determined as the melting point. This was done on Microsoft Excel and GraphPad Prism 9. Here we see the melting points, and the red indicates, again, the adeoxyguanine, and the blue, the abasic sites. It is accepted in the literature that a temperature difference of 2 degrees or more when compared to the control is significant in terms of stability for the oligonucleotide. So the main observation from this is that the abasic sites were excuse me, the abasic sites were much lower than the HCNA with the 8-oxoguanine. And this could possibly mean a lack of triplex formation in the presence of oxidative damage. The only exception to this was actually this second location with the 8-oxoguanine in the foldback. So some conclusions, 8-oxoguanine and abasic sites destabilize the HDNA, and HDNA in the presence of ROS might still get repaired by NER. Future studies can further analyze the mechanism of destabilization on HDNA due to modified bases. So I want to thank my research advisor, Dr. Zoel Foote, my summer research partner, Corinne Stein, the chemistry department, the Welch Foundation, and the NIH. So I am happy to answer any questions.
Let's see, Dr. Niemeyer.
Great job, Elijah. It's really interesting. I have a couple of questions for you. One, can we take a look at the melting point, not data, but the original graphs of the spectra? So one of the things I noticed, and I wonder if you can just comment on, so I know you take the first derivative in order to get the maximum, which is the inflection point, but I noticed the shapes of those, the melting points are actually different. And so does that mean, I seem to remember back somewhere that the shapes of those can tell you information about like how many species are present and other things, because it looks to me like the MCR2 has a different shape in that kind of tailing on the front, or that front end, than what I see in the OG1 and the AB1. So I'm wondering if that's something that y'all investigated, or if it's just that happens, or if it actually means something.
So I do know that we were looking at the thermal data at every 0.5 degrees Celsius. And so this actually created a really rough zigzag for the first derivative. And this graph has been smooth a couple times over. So I don't think it's exactly indicative. I think it speaks more on the machine and the way it was reading it, kind of bouncing back and forth. So it would go up like 1 degree, and then it would come back down a little bit and then go up. And so it kind of created a zigzag. So I don't think it indicates as much to the structure as it does the data collection, but studies weren't particularly done on that. Great observation, thank you.
So you think it's more like a noise phenomena than a?
I think so, because if I were to show you the original graph, it would just be kind of crazy. Now it could possibly indicate that it is structurally different. And again, on this graph, I only graphed OG1 and AB1, but it would be interesting to compare all 4 different locations and see, and maybe look into that.
Yeah, because I see the noise in there, but it still looks like on top of the noise, there's it's much less Gaussian in shape. None of them are Gaussian, but I noticed that one to a much greater extent has a, this kind of tailing leading up, as opposed to a little bit more symmetrical in shape. So
Elijah, what does it mean if it's a sharp curve versus like a very broad curve like Dr. Niemeyer is mentioning?
So that would here, I have a different graph.
Like just on that one, like you have a black one, right? Yeah.
So just based off of, like, I'll walk through this real quick. So we know that the triplex is over here at the bottom, and we know that it's being denatured either into the duplex or a single strand. So if it's a sharp curve, I would assume that this denaturing is happening rapidly. So maybe maybe it needs a higher energy amount, and then once it reaches that, it just immediately denatures. So it instead of a gradual energy increase that's slowly picking at it, it would just be sudden, possibly.
that makes sense. Thank you. Can I ask one other just quick question?
Yes,
can we look at that chromatogram that you had up, this was for the previous
Yes.
study.
This one?
Keep going. Previous, I think it was when you were talking about, yeah, what the heck is that? You say it's a DNA chromatogram. Like, what is it a chromatogram of, and like, how is that measured? And that's like, if that's a chromatogram, it's like the ugliest chromatogram I've ever seen in my life. So I'm just kind of curious, like, what's that mean?
So
Like,
this is, if we think about it, lasers are being pointed at the DNA sequence, right? And since this is an HDNA forming sequence, some of the bases do end up clumping up near one another. And so when we sent it off to sequence and then they came back, sometimes they couldn't read it. But this is probably because of just the shape of the oxidized guanines, probably. And it could also be a limitation of the APE plasmid editor. So, but yeah, great observation. And there were some parts, this is only one, which is early on in the sequence, but some of the peaks were even worse than this. And we just rendered the data, not useless, but I guess useless.
so is it like LC mass spec? Is that what they're doing there? Is that what the chromatogram is? Is that how they collected it?
I'm not too sure. I actually do not know exactly what kind of sequencing was done on it. Maybe Sanger sequencing. I'm not too sure.
Okay, so you were getting these chromatograms that were analyzed,
Yes.
and then you're feeding them into the.
I'm just doing the analysis.
Okay, so you're feeding them into a library, because obviously it's really complicated because there's all these different peaks and stuff, and they're all overlapping one another. And so the job of this global align blast thing is to deconvolute like how these different peaks relate to the original sequence. I mean, is that what's going on there?
So if we go back to this slide, we know which plasmids that we actually transfected and put in. So based off of that, we have that sequence perfectly. You know, that's our control, right? And so then when we after the experiment, the DNA was isolated, and then that was the experimental sequence. So yes, this top original sequence is the known plasmid that Rachel talked about a little bit. And then the experimental sequence is what was generated after it was isolated and then sent off.
Okay. I think I got that. Okay, thank you so much.
Okay, we have a couple more minutes, so let's see Dr. Massey.
Okay, it looks like there's just like some frequency, there's some oscillation frequency on top of that chromatogram. Anyway, okay, sorry, can you go back to like the part 1 data slides? That was a nice talk. Thank you, Elijah.
Thank you. Is it this one?
Yes. Okay, so it looks like there's both, right, like a increase in the number of mutations when there's the positive for the damage, right, as well as kind of a big change in which mutations are present. So do you have any idea of like why there is a decrease really in like the number of small deletion mutations? Are those being turned into something greater, or something happening with those in both the bDNA and HDNA?
Yeah, when we were looking at the sequence, it seemed like a lot of the small deletions that we saw previously were just simply part of this large deletion now. And so that part of the sequence was gone. And so that's what I would speculate. Instead of these smaller deletions that were happening in this data, they were just being excised completely as a bigger portion.
Okay. And then are there error bars on these measurements as far as like the overall change in number of mutations?
No, they're not on this one because it wasn't done in triplicate or no error bars were calculated for these.
Okay, thanks so much.
Okay, we have 1 minute left, so I think Dr. Wiegand can ask his question.
So I think this can be answered in 1 minute. How common is HDNA? If I looked at a cell and I looked at all the DNA in there, how many HDNAs would I find?
So we know that they are, as a whole, I'm not too sure, but we know that we know that the hotspots for the HDNA forming sequences are near like promoter regions. Let me switch to one of my slides. Yeah, so we know that they're frequently found in the promoter regions. And so that's where I would assume, and however many promoter regions there are, I know that one that is commonly looked at is the CMYC gene, but an exact number, I don't know off the top of my head. So.
Okay, like 1% or 10% or 50%, we don't know for sure.
I'm not too sure. Correct.
Okay,